Today we will continue our discussion about the therapeutic efficacy of CRISPR knock-in engineered human CAR T cells. So we were discussing about the results of their CRISPR knock-in engineered human CAR T cells, where they knock in this RGS16 or where they knock in this interleukin 2 at the just after the or just downstream of the start codon of this RGS16, since RGS16 is also a tumor specific transgene promoter. So we were discussing about these results, and we have seen that the high expressions of cytokines not only in the tumors but also in the blood and spleen. The results for the tumors are shown in this panel K, where we can see that uh, cytokine positive cells are around this 33% in the mock, 31% in the RGS16 knockout, and 42.9% in the RGS16 with interleukin 2. And one of the representative of the flow cytometer results are shown in the right hand side of the panel K, where we can see that this IFN gamma is increased compared to the mock and also compared to the RGS16, and similarly the TNF cytokines are also increased compared to the mock and RGS16. So once having validated their CRISPR knock-in engineered human CAR T cells, they wanted to check whether the promoter genes that they have identified, is RGS16 and NR4A2, works well in the human patient conditions or not. So for that, they first check the single cell RNA-seq data that are publicly available of the patients, and they check it for the multiple cancer cells for the expression of NR4A2 and RGS16 in the peripheral blood stream and also in the tumor stream. And they are able to see that in the tumor stream for both NR4A2 and RGS16, the expression levels of the this NR4A2 and RGS16s are high in the tumors compared to the blood. So indicating that this NR4A2 and RGS16s are more tumor specific transgene expressions. So once validating that their candidate genes are also shown to be tumor specific transgene expressions based on the sickle cell RNA-seq data of the tumors. So what they did is they took the patients T cells, or the patients who are affected with B cell lymphoma, those T cells, and transduced them with NR4A2 GFP expressions and RGS16 with GFP expressions. And what they are able to see in the case of non-stimulated conditions, for the NR4A2 the positive expression size very low, around 4.6, and for this RGS16 expression levels around 38.7. This could be the non-specific, you can say that. And when they are stimulated or stimulated with ovarian cancer 3, there is a significant increase in the GFP expressions to 62.5% for NR4A2 GFP expressed CAR T, and on the RGS16 GFP expressions also increased from 38.7 to 63.5%. And thereafter they have quantified these expression levels in the panel N, where we can see that there is a significant expressions and the increase is statistically significant for both NR4A2 and RGS16. And the fold change they have also shown in the panel O, where the fold change is more than 20 for NR4A2 and between 10 to 20 for this RGS16, and there are some fold changes around 5 or 6 also, but those fold changes are also statistically significant. And they also check the after validating the efficacies with GFP expressions, they check it for the system with to express the cytokines. So for NR4A2, they interleukin 12 combinations, they see a significant increase compared to the control and mock case, and for RGS16 also they see a significant increase compared to the control and RGS16 knockout cases. So these results are also in line with what they are expecting. So once they validated that their CRISPR engineered CAR T cell systems works in the patient T cells also. They wanted to compare it with the previously published method where they used a single transduce transductions to form a CAR T cells, and at the same time engineered the promoter sites. So they compared the retro-CAR where they used two different transductions, first for the antigen and second for this CRISPR knock-in at NR4A2 and RGS16, with this TRAC-CAR transduce system where they use single transductions for antigen expression and at the same time for NR4A2 and RGS16 expressions. And they are able to see the comparable results in both their retro-CAR system and this TRAC-CAR system. That is, in the non-stimulated conditions, both of them shows a less non-specific signals. In the activity conditions, the positive signals are almost similar. And when they check their TRAC-CAR or single transductions engineered CAR T cells for the in vivo studies, in the in vivo studies to check for the tumor areas or tumor suppressions, they are able to see that both of them perform similarly. That we have both perform in a similar manner that we have seen in the previous video for the retro-CAR engineered cells. That is, for the NR4A2 interleukin 12, the tumor is suppressed compared to the other control cases, and similarly for the RGS16 interleukin 2 case, you can see the suppression of the tumors compared to the other control conditions. So this is how they validated that their CRISPR knock-in engineered CAR T cell systems is can work on the patients towards diagnosing the tumors, especially the solid tumors. So that's all for this video, and this is the last main figure of the paper. If you need to understand more about these experiments and this topic or this paper in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.